Okay, there we are. Does anybody have any questions before we get started? So to reiterate, we're going to do the Zoom quiz Wednesday when we don't have, hopefully, don't have weather issues, internet connectivity, all the issues that are happening today. There's Brandy. By the way, is everybody doing okay where y'all are? Besides probably being a little bit drowned? Okay.
Yes.
Good. Eleni, you made it in the rain. Okay. Okay, well, we're going to get started. And let's see, I'm trying to get my screen up. So we've talked about photosynthesis. Ah, I got you. Let's see, we talked about, I don't think we started here, did we? I think we ended with the pigment slide. Is that correct? Does that sound familiar? Okay. So I wanted to pick up the photosynthesis as a whole. So we've got, hello, doggy.
Sorry about that.
That's okay. I'm fine with, you know, small animals or even large animals coming to class. I'm okay with that. If I could bring my dog, I would. They just won't let me bring him in the building. When I'm at home, I'll let y'all see him sometime. Okay, photosynthesis. Big deal because it's different than what you get in animals, plants, animals, fungi, bacteria, archaeans. So it's only going to be found in plants, the way we're going to look at it. So there is photosynthesis, we mentioned briefly in bacteria, but they do it a different way. There are algae that do photosynthesis, and they're going to be pretty much the same format of how we're doing it here because they are related. Okay, so let's get into this because this is something that we we do cover in other classes, but it is very pertinent, you know, to a botany course. So the purpose of photosynthesis is for energy, you know, that's required in cells, and it needs to be in the form of ATP. Plants make ATP using light as the energy source. Now, do they do it directly? Not exactly. So we start with photosynthesis. We create usually a carbohydrate, though it can be turned into other molecules. And then we do what normally most things do. We go through cellular respiration to make ATP. From that, we're going to call it food that the plants make. This takes place in the chloroplasts, and specifically those parts that are green on the plant are usually going to be the ones doing photosynthesis. I would like you to know this equation at the bottom, okay? It is 6 CO2s plus 12 H2Os and light is their energy source, gives us C6H12O6 plus 6O2 plus 6H2O. Okay, first off, what is C6H12O6? If you were to put a name to that, what molecule would it be called?
Glucose.
Glucose. Good job. I think Brandy said it too, didn't you? Saw you mouth it. It's definitely glucose. And it's an important molecule, okay? Why is glucose important? As animals, why do we consider it to be important? two really important words that are on the screen. One starts with a C, the second one starts with an R. Any guesses? Cellular respiration. Sound familiar? What is glucose in cellular respiration? Uh-oh, y'all got me worried. Okay. Are y'all just being really quiet, or do you really not know? Give me a heads up. What does cellular respiration and glucose have to do with one another?
I don't know.
So
I don't think.
okay. Actually, it's the other way around, Elena. Um, so instead of it being cellular respiration makes glucose, it is cellular respiration uses glucose. So if you look at the equation at the bottom here, actually it's photosynthesis that makes glucose or sugars. Respiration then takes those products of photosynthesis, goes through the process of cellular respiration, and outputs ATP. Okay. Now remember, nowhere along the process do we make energy. Energy is just being transferred from one thing to another. So it starts with light. Light energy is absorbed by photosynthesis. It gets incorporated into the bonds of things like glucose and those molecules. And then we take those glucose molecules and we input them in a different process in cellular respiration in the mitochondrion, also the cytosol. And you start breaking those bonds to release energy, to which you can then make ATP from the energy that you've released. We really want to make sure that you understand this. This is like fundamental to life on Earth. We have to have photosynthesis to be able to get energy into our ecosystem. And then from our ecosystem, everything that's alive will do some form of respiration. Usually, there are exceptions to that, but that's a big deal for most living things right now. Okay, now when we have a green part of the plant, that refers to probably that plant having chloroplasts in that region of the plant. Do all parts of the plant do photosynthesis? Are all parts of the plant green? No. Can you name some places of the plant, or parts of the plant, I should say, that don't do photosynthesis? Okay, the petals, okay. They may have at one point, and then they change from chloroplasts to chromoplasts, but while they're the color they are, probably they're not doing photosynthesis. The roots, exactly. The roots are a really obvious one because what's important about them? They're underground. Exactly. Okay. Does that make sense for everybody? What about the bark of a tree? Do you think it's photosynthetic? No, no. But when it's really young, if you'll look, a lot of times when it's really young, maybe in the first year of growth, it is kind of green on the stem, or maybe you'll find new growth of branches, and those might be green. So they might be photosynthetic, but later on when they cover over with bark, that cork cambium, it's not going to be photosynthetic. Okay, so keep that in mind because we've got to move photosynthesis around the plant. Okay, let's keep going. So we've got our C6H12O6 being produced in this. Notice that our inputs are carbon dioxide, water, and light. Where does the carbon dioxide come from? It's going to be the atmosphere around us, okay? How does it get there? Well, there's a number of ways. Most organisms that do respiration release carbon dioxide to the atmosphere, and then it gets taken in by the plants. We also release it from burning fossil fuels. So there are some other mechanisms. There we go. Of course, we know water is absorbed through the leaves. Okay. Make sure I'm getting messages. Pay attention to things today, especially. So here's a comparison of two different kinds of photosynthesis. The one that we usually see in plants is the one at the top. The one down here is a type of photosynthesis that is a little bit different. These are those bacteria that do it with the bacterial chlorophyll. Do you see the difference? What's a major difference in these two versions of photosynthetic reactions? We're looking for a molecule difference. Do you see it?
Is it the H2S on the left-hand side?
It's definitely the sulfur, actually. So there's H2
Okay.
on the left, and then if you look on the right, instead of having O2, you've got two sulfurs. Okay, so in this case, instead of having oxygen, you're having sulfur as a base atom in these molecules. So they don't use oxygen. They can actually do this process without oxygen in the being produced, I should say. And also the water, it's not water that they use H2S. So you're going to find these bacteria in very different environments than you would our regular plants. Okay, that's all I'm going to say about it, just to show you that these bacteria do things rather strangely, even though the process is really not that different. It's just the replacement of an oxygen with a sulfur. Okay, it seems to be that the bacterial form of doing photosynthesis is actually ancestral. So let's go back to that first phylogeny kind of information we talked about. So bacterial chlorophyll probably developed first and later became oxygenic, when the atmosphere became more full of oxygen. And instead of using regular like white light, which uses the colors of light, I shouldn't say uses, but white is all of the colors of light. Visible colors, I should say. Bacterial photosynthesis will use infrared light, which is really interesting to think about because infrared can penetrate further into the soil and things like that. They don't have to be on the surface to be able to absorb that. And I said it's far, but it does penetrate further. So no oxygen, and then you've got your sulfur, basically a gas in this case. So it's a very different. It's not different, and it's different in the same. Whereas when you get to the oxygenic photosynthesis, what's considered to be derived. Now remember, ancestral would have developed first, and then we derived something from that. So this would have come afterwards. So some mutation, some change, changed the bacterial chlorophyll into a chlorophyll-style molecule using blue and red light as the main sources, oxygen being produced, and then we've got water as our H plus donor. So, and of course, this uses things that are readily available on the surface of our Earth, which is, of course, where plants live. Major steps in photosynthesis. There are two major sets of photosynthesis reactions, okay? Light-dependent reactions, the first phase of photosynthesis, okay? Major step one, okay? There'll be major step one and major step two, if you want to think of it that way. Major step one, light-dependent. So what do you need for this one? Light. So what happens is it's initiated by photons that actually come down and bam, hit on the chlorophyll molecules, and they have those little antenna that kind of stick up and catch those photons. Photons come from light, by the way, or they're our light. Light's a strange thing, by the way. But photons have some interesting properties, but they're able to hit the chlorophyll molecules, and this causes the excitation of electrons in these areas, and they kind of get shot off and into the electron transport chain of this, which causes an absence of electrons. Then water happens to be around if everything's going well for the plant. And water molecules will actually get split apart into their electrons, their hydrogens, and their oxygen. And that electron that got lost, or electrons that got lost because it got hit by photons in the chlorophyll, they get replaced with electrons from the water molecules. So it creates this movement of electrons through an electron transport system. We'll produce some ATP, but this is not the ATP that's going to run the plant. This is going to be used in the next step of photosynthesis. We'll also produce something called NADPH. And both ATP and NADPH are considered to be electron carriers, which will take their electrons to the next step of photosynthesis. Okay, so hold on to that thought. Keep in mind that with chlorophyll, you'll see that there's a range in which it absorbs better here and here. So chlorophyll A is blue and red. Chlorophyll B is a little, you know, on this side of blue. But nothing's really in that green range. Okay, it's close, but it's not quite there. So we absorb in this kind of bimodal distribution of light. And if you take plant phys with me, you'll actually get to extract some chloroplasts and get to measure this. It's kind of neat. We used to do that in class. Every pigment, chlorophyll A, chlorophyll B, the carotenoids, xanthophylls, all of them have their own distinctive pattern of light absorption, except that green's basically not absorbed. Now, the second step of photosynthesis, so major step 2, is light-independent reactions. So light is not necessary for powering this one. And I'm being very particular in the way that I say this, because some people will say that it's light-independent. Light is unnecessary. That is absolutely not true. There are some reactions in this step that need light, but light does not power this step to work. What powers it? It's the ATP and the NADPH that we produced in major step 1. So what we see is we finish conversion of all those things that we've absorbed, basically turning light energy into chemical energy. Another term for the light independent reaction would also be called the Calvin cycle. So you could call this the Calvin cycle instead of the light independent reaction, and I would be totally fine for that. And I would actually prefer it, although I take both. So what happens here is that carbon dioxide comes to play. We haven't mentioned carbon dioxide in the first step, but carbon dioxide is absorbed and it gets combined with something called RUBP, or ribulose bisphosphate. And then it's combined with molecules that will eventually go through this process. It's a circular process that converts sugars converts things into sugars. So we're taking that carbon and basically attaching a bunch of carbons together. So carbon dioxide has 1 carbon. When we start attaching them together, we'll get a 3-carbon molecule, and then we'll get a 6-carbon molecule. And of course, glucose is a 6-carbon molecule. So we're creating bonds, storing the energy from light that we've absorbed and transferred through the ATP and NADPH into this set of reactions. So your light-dependent reaction has energy come in to the first photosystem in the chloroplast where the chlorophyll is at. Light hits it, it excites the electrons, and then it kind of works its way along, creating some ATP, and eventually will lose some energy, and then light hits it in photosystem 1. By the way, just so you know, the reason why 2 comes before 1 is because 1 was discovered first, and then they found a second one, but it happened to be before it in the process. So they're out of order in terms of numbers, but the 1 and 2 gives them discovery times. But we then power another electron, kind of boosted off of this with light in both of these photosystems. Water over here is giving us our electrons to refill and do it again. And we're basically pumping electrons through this, to an NADPH. We also have ATPs that are being produced back here. Not a lot though, very, very few. Then it goes to the light independent reaction. The NADPH comes into this, drops off some of its electrons, gets converted back to NADP, which can go back and pick up another electron. ATP does the same thing. It drops off its electrons and it gets converted back to ADP, which can go back over to the other side here, light-dependent reaction, and get more electrons and come back. I think of them as kind of taxis. They go, drop off, come back, pick up some more, go back, drop off, come back. Does that make sense as an idea? They're just there to pick them up, those electrons, and they drop them off, and then they go back and pick up some more, and then they drop them off. Once they drop off those electrons, when you combine it with the carbon dioxide, you turn them into 3 PGAs. There's a 3 carbon molecule now. So they took 3 carbons from the CO2, combined them together. We keep going around. We ended up with 2 more kind of slightly different 3 carbon molecules. ATP is continuing to drop off energy, and we end up with a 5 carbon sugar that will later get converted, which is the RUBP, which will later get converted into a 6 carbon combination. Now, this doesn't show it, but the excess in the process gets kicked off, and that's what becomes the food, the glucose, the sugar that the plant is going to use. Okay, so we can only do this if we make enough to make an extra amount, and then we kick it out, and that's what's going to be used by the plant to build itself and go off into cellular respiration and other things. Okay, that's the very quick and dirty photosynthesis right there. Do you have any questions on that before I go to the next part of photosynthesis? Give me a thumbs up either through your actual video or through Zoom. Is everybody okay?
Yes.
Okay, awesome. So let's pick up with the next one. Oops, trying to get to there. Okay, plants of the day. So this is a water lily in the Nymphaceae. This is it's called a Victoria lily, named after Queen Victoria, and it has the largest leaves that I'm aware of plants in the world, at least the largest single leaf that's not divided. There are maybe some other plants that have larger divided leaves. But isn't this fascinating? This little girl is actually sitting on a floating leaf in that water. It's so cool. I've seen a picture of a lady from the Victorian era standing on one of these. Now, I have a feeling she was probably rather small, because I know if I stood on one of these things, I would be wet. I would have gone through it. They don't allow you to do this in most places because they are still rather fragile. And I know of only one place relatively near to us that has these, if you ever do want to see them. They're not these large, though, where they have them. But the Birmingham Botanic Gardens, which is free if you ever want to go, completely free. It's beside the Birmingham Zoo, which is not free. They have some in the summer, and they have some ponds as you walk in to the right. You have to kind of go up some steps. Or a ramp, I can't remember which, maybe both. And they actually have some of these. They have a beautiful flower, okay, huge flower, which has no distinctive number of petals. It's hard to tell. There's no specific number of things. One of the things that indicates is that these flowers, these plants are rather old in the what do you call it, evolutionary development of flowering plants, because later on, later flowering plants develop specific numbers of petals. Okay, do y'all remember when we talked about how most animals have a distinctive number of fingers or other digits on their paws or hands or whatever? It's usually 5, or you can tell that they've had something changed from it. Well, in plants, you see this with their petals and other flower parts. And we're going to go into this in greater detail later in the semester, but just heads up on this. Y'all are all familiar with the magnolia flower too, right? That one's another one that doesn't have a distinctive number of petals or stamens or um, pistils and things like that. So it's another one, and it's old. Okay, let's look at this. Okay, I need to switch to, oh, let me stop the share. I want to switch to something else. Switch to a whiteboard. Okay, let's see if I can do this from memory. Because one of the things that I want you to do is to be able to draw out photosynthesis. So I'm going to do it so you can see me doing it from memory. So in photosynthesis, and we're going to keep this really simple. It takes place in a chloroplast, right? Okay. Somewhere along the way, there is a photosystem. It's going to be photosystem 2, embedded into the membrane. Okay, so keep that in mind. So remember, does anybody remember what the name of those membranes are called that all the photosynthesis takes place in? Starts with a T, TH. Thylakoid, sound familiar? Nope. Yes? Can I get any affirmation? We're good? Okay. Just want to make sure. Getting a little bit worried. Okay. So we said that there is electrons, okay, that get kicked off. When I say kicked off, it's because they get excited. And it's not because they're getting a birthday present or something. When I say they get excited, it's that there is something that hits these photosystems, which is light in this case, and it gives them energy. So their excitation levels, their energy levels get increased, and it kicks off electrons, well, leaving this without electrons. Okay, so while these electrons are going to move, and we're going to talk about them in just a second, how do we replace the electrons? Where do electrons come from to replace this? And we just went over it. I'll give you the first letter. Second letter, except it's a number. Can anybody give me the third one? Thank you, Kyra. Appreciate that. Yep, it's water. Okay, H2O. So we said that it breaks into its pieces. What are the three pieces that this can be broken into? Well, one of them obviously is the electron, right? What do you have left over in this H2O? If we take out some electrons, what do we have left over? It's in the molecular formula. Okay, we're gonna have to start guessing. At least somebody guess something. Okay, Carrie, we got hydrogen and oxygen. Okay, so those hydrogens are protons, right? Hydrogens are just protons if you take away their electrons. The electrons are over here, so they come in as protons, H plus, okay? So where does the oxygen go? It gets left, okay, just off into the atmosphere, especially when you've got two oxygens that are going to join together. When you have two water molecules broken and you end up with O2. And O2 is the kind of regular oxygen gas we've got in the atmosphere, right? Okay, so basically, is oxygen used in this process? Yes or no?
If it's left, I would say no.
Perfect, exactly. That's exactly what that means. So, isn't it funny that when people say, oh, photosynthesis is, all about making oxygen? No, it's not. Photosynthesis gets rid of oxygen as a waste product, okay? So you can tell that when we think about plants and the way they do photosynthesis, as an animal, we think about them in a very egotistical way, okay? We think about them, about what they can do for us. We even look at their waste as for us. Isn't that so nice that they give us oxygen? No, they're getting rid of it because they don't need it. Does that make sense? It's like worms looking at poop. Oh, look what that dog left me. I'm pretty sure the dog's just like, I need to poop. You see what I'm getting at, hopefully. Okay, now these electrons have to continue along. So this electron replaces these that got kicked off. The ones that got kicked off keep on going. There are some other steps. I'm not going to go into great detail right here because I don't really want you to draw this for me. I am going to mention cytochrome B as an important step, but it's not really driving the process. There is plastoquinone and some other things that are basically like stops along the way, but they're not the energy. All they do is they're stop, stop, stop. As the electron is losing energy as it moves, these are stops along the way. It's like three major stops. What I really am interested in you getting into is where does it go to pick up more energy again? It's going to be your photosystem 1. Well, guess how that electron gets excited again? Can anybody guess? It's going to be light, right? We're going to do this again. And the electrons keep getting kicked off. Now, where do they end up? They end up in NADP plus, and I left off my D, NADP plus, getting that electron. In fact, it's going to end up like this, NADP. That looks like a P too. D. It's hard to write with a mouse. Okay, H. Okay. So we've actually picked up a hydrogen and 2 electrons. So this has now become an electron carrier. And since I can't really draw very well or write really well, I'm going to type it in. Okay, electron carrier. Now, we said that you make NADPH. Do you remember what the other thing is that we make here? Starts with an A. Ends with a P.
ATP.
Awesome. Yes, it's ATP. Yeah. So a number of y'all got that. So we make ATP. Now, we don't have anywhere in this process showing where ATP gets made. And it's because there are lots of steps up here where more hydrogen comes in. Really awful drawing. This is even worse than what I have in real life. They get pushed through this series of, it's an enzyme basically. Does anybody know what the enzyme is that helps to make ATP by pumping H pluses through? It's really famous. It even has the ATP in its name. ATP synthase. Okay. So when these things come through, we end up with ADP plus inorganic, sorry, inorganic phosphate that's kind of hanging out around. Let's make an arrow here. There we go. That will create a ATP. So what's the difference between ADP and ATP? Besides the letter, what's those letters mean? Two instead of three, okay? So we've got ADP, which is di. This is adenosine diphosphate. There's two phosphates. When we add a third phosphate, it becomes triphosphate. Now, when you add a molecule, are you going to be storing energy or releasing energy?
Storing.
Storing, exactly. Okay, so we're storing when we make the ATP molecule. What about NADPH? Are we storing or releasing energy?
I'm sorry, I missed that. What was the question?
So we've got NADPH. Are we storing or releasing energy in this one?
Releasing?
Yeah.
You think it's releasing? Kyra says storing. So
Aren't you? I was gonna say, aren't you going from ATP to ADP there? Or
not up here. There's no ATP up here.
okay.
So we've got NADP plus, and now that's a plus being a positive thing there, and we get to add to that electrons. Oops, not the right. There we go. Does that help looking at it this way? We've got NADP plus electrons gives us NADPH.
So it'll be storing because you're adding electrons.
Exactly. And that's the thing I really want y'all to get here, okay? So when I'm asking questions for y'all, I'm not trying to torture you. I'm trying to help you to make connections. So that process we just went through went, oh yeah, we're storing. The full idea of light-dependent reaction, the first major step of photosynthesis, is all about storing energy. Where did that energy originally come from?
The sun?
Yeah, good job. Okay, the sun. Okay. And we've got two processes here in which sunlight energy becomes chemical energy. So we go light energy becoming chemical energy in this process of photosynthesis, especially at the points of the photosystems. Okay. Do y'all think y'all could recreate something like this for your light dependent reaction? This is just the first half of photosynthesis. Ikiria's like, I can't. Okay, good. I like that confidence.
I think just have to like watch you do it over again.
And I would really suggest practicing on your own. Like maybe watch this again and then turn away from it, pause it, draw it myself, come back and check. Okay. You could even turn it on again since y'all have the video, or you will have the video. Then you'll be able to go, okay, as I talk through it, you can either draw it out or you can draw it out and then see if you did everything that I'm saying. But I want you looking at yours just as much as you look at mine, because the process of you drawing is going to put it in your head in a different way than if you just watch me. Okay? Does that make sense for everybody?
Yes.
Okay. Whoops. Went a little too far there. There we go. I lost my screen share. Okay, let's go back. So photosystems 1 and 2 are involved in photosynthesis. Keep in mind that they're found in the thylakoid. Okay, I'm going to go back because I want to show you something else. Let's see if we can. Okay, great, it doesn't disappear. Let's add two things to this. This membrane here, let's see, where's the best way to draw this? Okay, let's do this way. What is the membrane? So it's the membrane bound process. And let's say this way. Membrane is called what? What did we say the membrane is called? Starts with a T.
Something. Is
Thy,
that right? The lake. The lake. The lake.
thylakoid. Thylakoid.
I could see it, but I couldn't figure out how to pronounce it.
It's just 3 syllables. Thylakoid. Okay, thylakoid. Now, what about the space in between where these hydrogens are going in, these H pluses? Okay. In fact, I'm sorry, this is not right. Let me erase this. Where do I erase? Where's my eraser? So actually, this is not going to be inside here. There's actually going to be another set of membranes outside. I'm not going to draw it on here because it's not relevant to the light dependent reaction, but I just want to see what is not membrane. The space in and around where the second half of photosynthesis takes place, where does it take place? Or what do you call that area? Starts with an S. T.
Stroma.
Stroma. Okay, so this is going to be kind of like a cytosol style region outside of the thylakoid membranes. Okay, now not involved in this process, so I'm going to delete it. I don't want that there. Okay. Let's see. So these extra H pluses are going to get moved around in different places, and they're going to get kicked into the stroma and other things to like make ATP and be used in other places. So we've kind of talked about that. Okay, let's move on. So let's look at something you're going to see under the microscope whenever we get to have lab again, because isn't this fun? Next week we don't have class on Monday and a lab because we have holiday. So we've got like 2 weeks before we're back in lab again. You okay with that? Probably okay with that. So we'll remind you of things you're going to do before then. So we'll have to play some catch up since we've missed some stuff today. But here we go. This is a leaf that has a lot of chloroplasts. Okay. Now this specific one has chloroplasts very much around the vascular bundles, but you'll also see smaller chloroplasts in and around these other mesophyll cells. So definitely that's what you're seeing. Photosynthesis comes through, I'm sorry, light comes through and photosynthesis takes place where you see green. Okay. So we've talked about how green is the color that is not used in photosynthesis. And it's because, again, that we do not have absorption in that green area. Okay, so right in this area right there, kind of where that line kind of lines up. We get absorption before it, we get absorption after it, but we don't get green. there was a really cool experiment that was done to show this. I forget the guy's name off the top of my head now. What he took was a green strand of filamentous algae, and he took a prism under a microscope, and he shone different colors of light. He took a prism to break white light into its different bands to see what colors of light are used. And what he found is that when he put oxygen requiring bacteria near this algae, of course, they're going to go where the oxygen is being released, right? And so what they did is they kind of grouped up where the most oxygen was being released. And guess what? It's in the blue. It's in the blue and red range. And right here in this middle area, basically not being used at all. Isn't that a cool experiment? Like, how simple is that? You just look at it under the microscope and see where the bacteria go. Totally wished all the easy experiments like this were not done already. But this is cool. So we've got two types of photosynthetic units. We've got two photosystems. Remember, the photosystem is where you find the molecule, the pigment, which is the not just chlorophyll, but the other accessory pigments too. They are involved in making ATP because they are helping to push hydrogens into them, into the inner parts of the membrane, and then it gets pushed out through an ATP synthase. Only organisms that have both photosystem 1 and 2 can produce NADPH and oxygen in this process of creating electron flow. Okay, there are some other organisms, I think, that have one or the other, but if you don't have both, you don't get both NADPH and oxygen as a consequence, which is a good thing for us. So photosystems are found in leaves or any other, chlorophyll containing tissue, such as the leaf inside of it has mesophyll cells, which have chloroplasts, double membrane. And then inside you've got the thylakoid membranes. And then around that's the stroma, but you can see inside the thylakoid membrane, you've got embedded molecules with these little antenna-like things, which are the pigments. Okay. Now, please be careful when I say the word pigment. I have a feeling that your brain is probably thinking color. Every time I say pigment, right? You're probably thinking like it's a paint. That's not the idea that we're trying to get here. A pigment in biology is a molecule that captures a certain color of light. Now, that gives them color, but that doesn't mean that they themselves are color. So we don't want to think, oh, a pigment's just green. That's how we think of it when we're a painter, but not as a biologist. The process is still somewhat similar in how light works. If you see a chlorophyll molecule and it's green, what colors are being absorbed and what color is not being absorbed? Well, everything but green is being absorbed and green is not. I just saw that it's 8:52. I've been talking too long. Guys, you got to stop me, okay? Let me stop right there. I'm going to stop the recording.